In topic 3.2, we learned about genes, what they are, how they affect organisms, what leads to genes changing, and what a genome means for a species. In topic 3.2, we will take a closer look at chromosomes, which is where genes are found. We will learn that chromosomes carry genes in a linear sequence that is shared by members of a species. To begin this investigation, we will look at prokaryotic cells and how chromosomes are stored. In prokaryotic cells, we see that they only have one chromosome that consists of a circular DNA molecule. This chromosome is suspended in cytoplasm and free to move about within the cell. The area where the chromosome is found in a prokaryotic cell is referred to as a nucleoid. Prokaryotic chromosomes are free of proteins, unlike eukaryotic chromosomes. This lack of proteins in prokaryotic cells is essential for allowing the large chromosome to fit within the small prokaryotic cell. The presence of protein would allow there to be a structure to the chromosome, but a lack of protein structures allows prokaryotic cell DNA to be supercoiled, which just means to be twisted into a small and compact shape. This small and compact shape fits comfortably within the cell, even though the chromosome strand is much longer than the cell itself. In addition to this singular chromosome in prokaryotic cells, some prokaryotic cells also have plasmids. Plasmids are small circular DNA molecules that contain only a few genes and are capable of self-replication. They are not naturally found in eukaryotic cells. Plasmids allow the sharing of genetic material between bacterial cells. This sharing of genetic material can allow bacteria to evolve within a generation. The exchange occurs through a process called conjugation, where the sex pili of the bacteria with the plasmid links with another bacteria cell. The plasmid is copied within the first cell and passed to the second cell. Plasmids are essential in bacteria's building immunity to antibacterial drugs. There are usually one of two plasmids per chromosome in prokaryotic cells, but for some smaller plasmids, the number can be up to 50. The number of plasmids within the prokaryotic cell has been shown to influence the strength of the plasmid-borne characteristic. This is especially true for antibacterial resistance. The structure of eukaryotic cells differs from prokaryotic because of the proteins used as a structure for DNA in eukaryotic cells. Eukaryotic chromosomes are linear DNA molecules associated with histone proteins. The histone proteins create a compact structure for more efficient storage of DNA. The structure and organization of eukaryotic chromosomes can be broken into 5 levels. Nucleosomes are formed from DNA that is complexed with 8 histone proteins. Chromatosomes are then formed from a linking of one histone protein to the nucleosomes. Solenoid structures are then formed from the coiling of the chromatosomes. This gets condensed into fibers about the size of 30 nanometers. Chromatin is formed from these fibers, which are now loops, which have been compressed and folded around a protein scaffold. Chromatin will then supercoil during cell division to form chromosomes that are visible under a microscope. Genes are located over many different chromosomes in eukaryotic species. When chromosomes are compacted during cell division, we see the shape of chromosomes appear. The shape that forms is due to the chromosome being constricted in the middle, called a centromere. This splits the chromosome into 2 arms. The shorter of the 2 arms is the P arm, and the longer section is the Q arm. The location of the centromere and the length of each of the chromosomes and their respective arms are unique to each eukaryotic species. To investigate the structure of chromosomes, we use staining as a method to show the bands of each chromosome. The staining works by showing where there are rich areas of adenine and thymine base pairs. This is where we see the stain the darkest. This is also where we see genes to be relatively weak. The lighter parts of the staining indicate less condensed chromatin where guanine and cytosine are rich. Through the staining process, each chromosome can be mapped to the genes it carries. Remember from 3.1 that each gene is found on a chromosome with its own location known as its locus. This locus is then mapped using a 3-step process. First, reference which chromosome the gene is located with a number or sometimes a letter. Second, reference whether it is on the P arm or the Q arm with a corresponding letter. Third, reference the G band location with a corresponding number. Organisms inherit their chromosome pairs from their parents. Each parent donates one chromosome of the pair. These chromosome pairs are homologous chromosomes, which means each chromosome contains the same genes but not necessarily the same alleles. Remember from 3.1, alleles are a very specific form of a gene. These chromosomes are homologous because they share similar structural features such as same size, same banding patterns, and same centromere positions. They also share the same genes at the same loci locations. Cells that have these chromosome pairs are called diploid cells. Diploid cells are present in most plants and animals. All somatic cells are diploid cells, with new diploid cells being generated through mitosis. Each diploid cell will possess 2 copies of a gene. However, in order to reproduce, the organism must produce sex cells that only have one of the pair of the chromosomes. These sex cells are known as haploids. Haploids only contain 1 copy of each gene. All sex cells in organisms are haploids, and they derive themselves from diploid cells through the process of meiosis. The fusing of sex cells or haploids is what is necessary to create a diploid cell, specifically known as a zygote, which can then copy itself and develop into an organism. There are 2 types of chromosomes, heterosomes and autosomes. In humans, sex is determined by a pair of sex chromosomes. These sex chromosomes are heterosomes. A human female possesses a chromosome pair of XX, where both of these chromosomes have genes that code for female sex characteristics. Males, on the other hand, have 1 X chromosome which is inherited from their mother, and 1 Y chromosome that is shorter than the X chromosome and is inherited from their father. The presence of the Y chromosome in a human is what leads to the human developing male sex characteristics. Because 2 haploids combine from your parents, if the sperm cell from your father is carrying the X chromosome, the resulting offspring will be a female. If, however, the sperm from the father is carrying a Y chromosome, the resulting offspring will be a male. This means that it is the haploid from the father that determines the sex of the offspring. In all cases, the female egg will carry an X chromosome. The remaining chromosomes that do not determine your sex are called autosomes. When trying to organize the chromosome of a species, a model must be present. The model developed to organize chromosomes is called a karyogram. This model shows homologous pairs in descending length order, with sex chromosomes shown at the end. To determine a karyogram of a species, cells are harvested, made to replicate through chemical means, then halted in the mitosis stage when chromosomes are condensed. The chromosomes are then stained, photographed, and then arranged according to the karyogram model. Karyograms are used to determine the sex of humans and if that human might have Down syndrome. Before the baby is born, the karyogram can be created and analyzed for abnormalities. To determine the sex of the baby, the sex chromosome must be identified. To identify Down syndrome, the karyogram will show 3 copies of chromosome 21. This abnormality will lead to delays in physical and mental development for the child. Take a look at the following karyogram. What is the sex of this person? If you said male, you are correct. We can see that the last chromosomes in the karyogram show a long X chromosome and a shorter Y chromosome. This indicates that the person is genetically male. One important characteristic that separates species genetically are the number of chromosomes in a species' genomes. The number of chromosomes is a characteristic feature of members of a particular species. This means that species with different diploid chromosome numbers will not produce viable offspring. There are some cases where species of different diploid numbers can reproduce, such as a horse and a donkey who can create a mule. However, the offspring will be left unable to reproduce, and this by definition means that the horses and donkeys are considered separate species even though they can reproduce. A second feature of knowing the number of chromosomes of species is that they can be compared to one another. When comparing the number of diploid chromosomes, however, we see that there is little evidence that a greater number of chromosomes means the organisms will be more complex. Take for instance the comparison of diploid chromosomes of humans compared to dogs. Dogs have 78 diploid chromosomes, while humans only have 46 chromosomes. While dogs are great creatures, they are not more complex than humans, and this shows that the number of diploid chromosomes does not indicate a more complex organism. Lastly, genome size also does not show a valid indication of genetic complexity. When comparing the genome of a human to a canopy plant, we see the canopy plant has 50 times more base pairs than humans, but canopy plants are not 50 times more complex than humans. There are, however, some general trends in genome size that can be found. Viruses and bacteria tend to have very small genomes. Prokaryotes typically have smaller genomes than eukaryotes. And sizes of plant genomes can vary dramatically due to the capacity for plant species to self-fertilize and become polyploid. One difficulty in the development of chromosomal knowledge revolved around the length of chromosomes. In the previous technique mentioned in developing karyograms, scientists interrupt the process of mitosis to investigate chromosomes. At this point in mitosis, the chromosomes have supercoiled, making it difficult to measure the length due to their compact nature. That's when scientist John Cairns developed his method for photographing chromosomes while they are uncoiled. This method is called Autoradiography. This process works by using cells that are grown in radioactive thymidine. This chemical reacts with the chromosomes and makes them radioactive. The radioactive chromosomes are then placed on a photographic plate and placed in silver bromide solution. The silver bromide solution reacts with the radioactive chromosomes and causes the silver in the solution to turn into an insoluble substance, which can then be viewed through an electron microscope. The visual through the electron microscope shows the insoluble silver that has reacted as little black dots. With a clearer visual of uncoiled chromosomes, the length can be measured.